Welcome to The Explainer. Today, we're diving into one of the most fascinating and, frankly, most contradictory signaling molecules in ocular physiology. We're talking about nitric oxide, or NO. Now, it's an incredibly tiny molecule, but as we're going to see, it holds the absolute keys to your vision. It acts as a critical protector that keeps the tissues of your eyes nourished, but under the wrong circumstances, it can morph into a destroyer that leads to irreversible damage. It really is a profound paradox. I mean, think about it. How does a fleeting gas, a molecule made up of just one single nitrogen atom bonded to one single oxygen atom, end up with total control over the complex blood flow in your eyes? It defies almost everything we think we know about how the human body communicates. We usually think of complex proteins and these intricate physical receptors, right? Not a simple, invisible puff of gas dictating whether your optic nerve survives or starves. All right, let's dive into this. Here is our roadmap for today. 1. Meet the unusual messenger. 2. Nitric oxide synthesis. 3. Mechanisms of vasodilation. 4. Danger of oxidative stress. 5. The link to glaucoma. And 6. Future eye therapies. Section 1. Meet the unusual messenger. Let's meet the molecule. So nitric oxide is a free radical, and it behaves completely unlike most biological messengers we study. First off, it's a gas, which means it's highly diffusible. It spreads out rapidly the exact moment it's created. It's also lipid-soluble, and its lifespan is extremely short-lived. We're talking mere seconds before it breaks down entirely. But its true superpower? That lies in its incredibly small size and lipid solubility. Nitric oxide completely bypasses traditional membrane receptors. It doesn't need a chemical key to unlock a door to enter the cell. It acts like a VIP waltzing right past the bouncers. It passes directly through cell membranes to communicate instantly with vascular endothelial cells, smooth muscle, and neurons. Section 2. Nitric oxide synthesis. Let's look at how it is made. The really crucial point to understand here is that because this free radical is so short-lived, it doesn't just spontaneously appear out of thin air. And your body can't exactly stockpile it in some biological warehouse for later use, either. It has to be actively produced on demand. It's synthesized from the amino acid L-arginine in the presence of oxygen by a highly specialized family of enzymes known as nitric oxide synthesis, or NOS. So when the eye needs to dilate a blood vessel, it literally has to fire up these local enzymes immediately. Think of these as 3 major factories or isoforms of NOS enzymes working in the body. Let's walk through them conceptually. We've got NOS1, the neuronal type, and NOS3, the endothelial type. Both of those are entirely calcium-dependent to function. They rely on normal, healthy calcium signaling inside our cells. But nestled right between them is NOS2, the inducible form, or iNOS. And this one is entirely unique. It's calcium-independent and typically activates only during times of cellular stress and inflammation. Definitely keep that inducible enzyme in the back of your mind, because that specific inflammation trigger becomes incredibly dangerous when we talk about eye diseases in just a bit. Focusing specifically on NOS3, or eNOS, which is found in the vascular endothelium, we find that this enzyme is constitutively expressed. That essentially means it's continuously present, just humming quietly in the background to generate a basal, resting level of nitric oxide that keeps blood vessels slightly relaxed at all times. But its production really ramps up when stimulated by specific triggers that spike intracellular calcium. Those triggers include biochemicals like acetylcholine and bradykinin, but amazingly, they also include physical forces. The literal mechanical shear stress of blood flowing forcefully against the vessel walls triggers eNOS to produce more nitric oxide, widening the vessel to accommodate the flow. Section 3. Mechanisms of vasodilation, regulating blood flow. Your retinal tissue has one of the highest metabolic demands in your entire body. For you to process visual information, your eyes consume massive amounts of energy. That means precise, second-by-second regulation of the blood supply is absolutely critical. And the biological cascade that makes this happen is just elegant. First, nitric oxide diffuses rapidly out of the endothelial cells lining the blood vessel, right into the adjacent smooth muscle. Second, it binds to and activates an enzyme called guanylate cyclase. Third, that activation triggers a massive increase in the production of a secondary messenger called intracellular cGMP. Finally, this rising tide of cGMP acts as a chemical switch that forces the smooth muscle fibers to stop contracting. They relax, causing the blood vessels to widen or dilate, which dramatically increases the flow of oxygen-rich blood. And this brilliantly illustrates a crucial physiological concept called neurovascular coupling. Think of it as a flawless, localized supply and demand system. It's the increase in local blood flow that perfectly matches increased neuronal activity. Basically, when you're reading, driving, or navigating a bright room, your retinal neurons are working harder and firing rapidly. Nitric oxide is the localized messenger that instantly senses this demand and dilates the nearby microvessels. It ensures that necessary oxygen and nutrients are delivered exactly where they're needed at the exact millisecond they're needed without oversupplying resting areas of the eye. Now, maintaining that normal ocular perfusion requires a continuous biological tug of war. On one side, we have our powerful vasodilators like nitric oxide and prostacyclin, constantly pulling to open vessels up and ensure maximum flow. Pushing against them are vasoconstrictors like endothelins and angiotensin, which actively narrow the vessels to maintain blood pressure and vascular tone. It's a highly delicate equilibrium. If you were to artificially inhibit that basal nitric oxide production we talked about earlier, you'd essentially just hand the rope over to the vasoconstrictors. This triggers massive vessel constriction and drastically reduces vital blood flow to the ciliary vessels and the optic nerve, literally starving the eye of oxygen. Section 4. Danger of oxidative stress, when NO turns harmful. So here comes the plot twist in our explainer. Let's contrast the two faces of nitric oxide. On one side, we have the protector. Under healthy, normal conditions, it promotes vasodilation, maintains robust retinal perfusion, and supports that brilliant neurovascular coupling we just explored. But on the other side, we have the villain. When nitric oxide encounters the wrong physiological environment, specifically a state of oxidative stress, where there are excessive reactive oxygen species roaming the tissue, it turns deadly. It combines with these unstable molecules to form highly toxic peroxynitrate, leading directly to serious tissue injury and cell death. The chemistry behind that toxic transformation is alarmingly simple and shockingly fast. When our protective nitric oxide reacts with something called the superoxide anion, which is a highly dangerous oxygen-free radical generated during periods of intense cellular stress or disease, it creates peroxynitrite. This new compound, abbreviated as ONOO-, is no longer a helpful signaling gas. It's a rapid, deadly chemical combination that absolutely wreaks havoc on the surrounding ocular anatomy. And peroxynitrosite is incredibly destructive. Because it's highly reactive, it damages ocular tissues at a fundamental, cellular level. It attacks structural proteins through a process called nitrosylation, fundamentally altering their shape so they can no longer do their biological jobs. It aggressively oxidizes lipids, literally tearing apart the protective cell membranes of neurons. It can even penetrate the nucleus to damage the DNA itself. Ultimately, this isn't just a temporary irritation. This represents a severe and often irreversible threat to neuronal survival. It's the molecular equivalent of a wrecking ball swinging inside the fragile tissues of the eye. Section 5. The link to glaucoma. Impaired autoregulation. The crucial takeaway here is how this molecular biology directly applies to glaucoma, which is one of the most common blinding diseases in the world. Normally, healthy eyes smoothly autoregulate their blood flow, keeping the optic nerve nourished even if a person's systemic blood pressure or intraocular pressure fluctuates throughout the day. But that vital autoregulation is heavily impaired in patients suffering from normal tension and primary open-angle glaucoma. Nitric oxide is supposed to regulate that optic nerve head blood flow and maintain healthy vascular tone. However, remember that unique NOS2 enzyme from earlier, the inducible one? Well, when an eye is suffering from chronic inflammation, INOS kicks into overdrive, pumping out excess nitric oxide. In the presence of oxidative stress, this flood of NO converts into toxic peroxynitrite, actively worsening the neuronal injury and accelerating vision loss. In fact, cutting-edge experimental models have shown that if we can specifically inhibit that inflammatory INOS enzyme, we actually see notable neuroprotective effects. Section 6. Future eye therapies. Targeted strategies. Modern glaucoma therapies are increasingly recognizing a major paradigm shift. Managing vascular physiology is just as important as the traditional method of simply lowering intraocular pressure. But because nitric oxide possesses both protective and profoundly harmful properties, our therapeutic strategies have to be incredibly precise. We can't just block it all, and we can't just flood the eye with it either. Future targets are focusing on using controlled nitric oxide donors, essentially eye drops that release NO to actively improve ocular perfusion in starving tissues. Simultaneously, researchers are looking at selectively inhibiting the inflammatory INOS, shutting down the toxic factory without stopping the helpful baseline flow from eNOS. Add to that the active reduction of overall oxidative stress and the enhancement of general endothelial function, and we've got a comprehensive strategy.
It's all about restoring microscopic balance, not just wiping the slate clean.
This quote from the research perfectly captures the magnitude of what we're dealing with. Nitric oxide occupies a central position in modern ophthalmic science. It is a remarkably small molecule, literally just 2 atoms, yet its profound impact influences nearly every single aspect of ocular vascular biology. It's responsible for keeping our high-definition retinal tissues fed and communicating smoothly, while unfortunately also participating heavily in the very disease processes that threaten to take our sight away in the first place. Which leaves us with this final thought. We know nitric oxide is a biological mastermind, walking a razor's edge between protecting the eye and destroying it. But as modern medicine learns to suppress its dark inflammatory side while simultaneously boosting its protective, blood flow enhancing benefits, will nitric oxide hold the key to the next great glaucoma cure? It is undeniably one of our most promising targets for preserving retinal and optic nerve function for millions of people. Thanks for joining me on this explainer, and keep questioning how the smallest molecules can make the biggest impact.